Good evening, everyone. We are waiting for all the participants to join. We'll be starting the session in next 5 minutes. Good evening, everyone. Myself, Divya. So, before I start, I'm thoroughly pleased to witness you all here in this webinar. It's my privilege to address you all on the behalf of our organization, Dollar Education. I welcome all the participants to the webinar. We're truly delighted by your presence. We all have gathered here today to attend this webinar on game changer machine learning methods in biology. We, as a company, okay, my slide is not changing. Give me 1 minute. I hope my screen is visible to all. I'm so sorry. There is some issue with my system. So, we as a company aim to we as a company aim to achieve great heights. This webinar intends to resolve the practical hurdles faced during the development of various bioinformatics software and algorithms used in biology. Our organization, Dollar Education, is an initiative to offer practical training on emerging concepts by making them affordable for all and develop a consistent environment of growing together in the field. We want to promote bioinformatics and make everyone to know the significance in the field of biology. The concept is just to connect and share ideas amongst various groups. You can find us on social media with Dollar Education to keep us all in touch and also share ideas amongst each other. We named our organization because of its multifaceted meaning. That is, it is the currency name in around 20 countries. It is popular as an affordable symbol and also used as a variable in computer languages. We want all our participants to get best out of our workshops and webinars. You can We make this all the sessions interactive, like you can post your questions and reply to the question being asked. We aim to provide source of each and every information with proper references, and it will be your next career jump to appreciate diversity and multiculturalism. Also, through our workshops and webinars, students understand the fundamentals of the field, and the researchers during the session they can apply these ideas to your projects. We have received over 150 registration over the globe, and from the record we can see that most of them are graduates or postgraduates, so they're in their budding age and they can get more out of this. The theme of our webinar is on applications of machine learning methods in biology. We have seen a lot in past 2 years, and it ultimately demands the knowledge of bioinformatics and machine learning in some perspectives. So for our today's session, we have Dr. Sandeep Dhanda and I would like to welcome him to our inauguration of Luminaire program. Dr. Dhanda completed his bachelor's from Kurukshetra University Then he earned his master's degree in science from MD University in Rohtak City. He then moved to Chandigarh and earned his PhD degree in bioinformatics from a joint program of Institute of Microbial Technology, Chandigarh, and Jawaharlal Nehru University, New Delhi. Next, he pursued interest and received postdoctoral training from La Jolla Institute for Immunology, San Diego, California, USA. Currently, he is working as a research scientist at St. Jude Children's Research Hospital in Memphis, City of Tennessee, USA. He's a well-known reviewer in for many journals, and he's also having the outstanding record of scientific publication and serves as a academic editor for several reputed journals. Dr. Danda published more than 50 articles in high impact journals and has developed dozens of heavily cited tools for vaccine designing and in the field of machine learning. He's an academic mentor in various reputed journals and a scientific advisor in Chiromic Biopharma from June 2020 till date. He is also a visiting lecturer in San Diego State University since 2016, and recently he completed over 1,000 citation of his research work in 2021. Today he's going to share his experience in machine learning methods for biology. Now I would like to ask Sir to take over the session. Sir, you can can you share your screen?
Yeah, thank you, Divya, for the kind introduction. And I hope my screen is visible and I can go and get started.
Yes, sir, it's visible.
All right, thank you. So I welcome all the participants for this webinar, and I'm going to share my experience about machine learning methods. And I have picked a couple of methods that I would call as game changer, and we'll go through why these methods are game changer, how these methods are being determined, and what purpose are they actually serving. So before going deep into the every aspect of machine learning method, let's briefly say our motivation. And I would like to thank Dollar Education for organizing this webinar. So these are some of the quotes to set the tone of this kind of presentation. So as Andrew next, he said a couple of years ago, just like the electricity transformed everything hundreds years ago, we are going to see the same thing in our industry because of artificial intelligence, and that's currently happening. And we are going to take some examples that actually justify this statement. And another statement I have picked is from Larry Page, who is the co-founder of Google. He said that artificial intelligence would be the ultimate version of Google, so that you can just speak to a search engine and you can get the answer what actually you're looking for. So we are getting closer and closer to achieve that goal every year. And the similar kind of statement is given by Mark Cuban, who is a well-known kind of investor and entrepreneur. He said like, artificial intelligence, deep learning, or machine learning, or whatever you call it, it is because learn it if you can learn it quickly. Otherwise, you are going to be dinosaur within 3 years. Though I mean, it's kind of an exaggeration in that statement, but still, it's making a point that machine learning is really, really very important, and it's time to learn it. So when we're talking about machine learning or artificial intelligence, I would like to start with a brief definition. The definition is here: the machine learning is an application of artificial intelligence. So machine learning is kind of application part of it that provides the system ability to automatically learn and improve from experience without being explicitly programmed. So that means we are learning from given example without every time telling this to be explicitly programmed for every single instance. Machine learning focuses on the development of computer program that can access data and use it to learn for themselves. So there are also some programs which can learn by themselves, and we will go through these different types of machine learning methods in our upcoming slides. So this is the source of this definition. So kind of the takeout keywords from this definition is this is an application. So we are going to apply this in biology. The other people are applying in other fields. So this is one of the application of artificial intelligence. And the second keyword here to take home is ability to learn automatically. So it has the ability to learn the things automatically. So that you provide the set of examples, it can learn from them, or they can also provide the endpoint, and it is try to find a path to reach that endpoint. And again, we should stress upon here, like this is a computer program. So everything when we taken together, this makes the definition of artificial intelligence or machine learning. So let's take some example that we are seeing today. So that's currently happening. The one example is Tesla. You have seen that these are self-driving cars running around the roads in many countries. This is because they have kind of learned the system to run the car by themselves. So this is self-driving car is one of the feature of the artificial intelligence. The other thing that you have seen in your life is like Google Assistant, Siri, or Alexa, where you just give some voice command and it can return you some kind of output based upon the information that already been fed in the system. And the other thing that you are seeing right now as an application of this AI is in Netflix or Amazon Prime. So whenever you are streaming some video, it automatically suggests you might be interested in this one, or you might be interested in that one. So those kind of things that trying to capture your interest and suggesting you the next video or whatever the content is available on that website according to the information that is already been given to them. So those kinds of applications are there in streaming platforms. The other application that you are seeing right now is LinkedIn or Facebook in social networking, kind of suggesting you might be interested in this post, you might be interested in this event, or you might be would like to connect with this group of people. So all those things are based upon the kind of reactions that we are giving on Facebook or LinkedIn or on any social media platform. So they try to capture this is our pattern, and from that pattern they suggest the next thing that we would be interested in. And these are the applications from different fields. And since this webinar is more focused or more aligned about talking about machine learning application in healthcare, so DeepMind is one of the things that's really changing the scenario of protein structure prediction. And we are going to talk about many other methods in healthcare or in biology that are changing the game. And let's talk about how different type of machine learning algorithms are. So if we take all the algorithms together, we can categorize them into three different parts. Either they are unsupervised, or they are supervised, or reinforcement. So unsupervised, we are not providing and the data label as an outcome, but we are trying to see if we have this data, how many different groups would be there. So those kinds of methods or algorithms are called unsupervised, because we are not providing the ultimate label to make it biased to make prediction along those lines. The next is supervised. The supervised something when we know this is going to be the outcome, and I'll give example of each of them in following slides. So the supervised is when we are giving data labels, and from those data labels we are trying to find out, based upon the information provided, can you please find which sample belongs to which class. So we are already providing classes here, that's why it's called supervised. And the reinforcements, we are not providing the labels as such, but we are providing an endpoint, and we are trying to track how it achieves from the start to the end point. So those kind of, and we have a system of rewarding for each correct step, so that every time it's try to move toward that endpoint, it's being rewarded, and it's being penalized when it moves in the other direction. So those kind of things are there in reinforcement learning. So machine learnings are broadly categorized into these three different classes. Talking about unsupervised in a little bit more detail. So this is kind of understanding the underlying pattern without being explicitly that divide this data into these many classes. So here we are not giving any labels. So the labeling is kind of the key point here, that there are no label given to the subject in unsupervised learning. And outcome is in the form of cluster. So how many groups are there when you just supply the data? Please find how many total number of groups are there in this data. So those kind of clustering algorithm or the data sorting algorithms are kind of classified as unsupervised learning.
Thank you.
The next I would pick is supervised learning. Here we are showing them this data is labeled. There are these many groups here. There are these many kind of classes here, and we wanted to see how, based upon this information, you are grouping this into those classes. And here to build this kind of algorithm, data need to be partitioned into train and test, and then kind of run on internal cross validation first. And from that internal cross validation, we also have some data taken out as an independent set, then try to see how we are going to perform based upon the data that we have taken outside. And it's always better to have kind of validation data in addition to independent data, so that we can run more and more validation, and we can rely more on the model that we are building. It can even be further classified, like the supervised learning could be just a classification. It could be two class, binary class, or it could be multi-class, or it could be regression. The regression means when we are interested in finding out a real number instead of different classes. Let's take that with an example here, the regression and classification. So when we wanted to see what is going to the temperature tomorrow, so it's going to be like regression model, because we are looking for a real number like 84 degrees Celsius or 84 degree Fahrenheit. In this case, like we are looking for a real number, that's a number here, the 84. But in other case, it could be classification. When we ask the question differently, will it be cold or hot tomorrow? In this case, this regression model is kind of divided into two different arms, and the one is we call as cold, another we call as hot. So based upon a cutoff, the the regression model are kind of trained also as a classifier when we wanted to see different classes, and these classes are kind of categorized based upon a cutoff. So less than this we are calling as cold, more than this we are calling as hot. So this is kind of dividing that real number into two different classes here. And let's take more example about the supervised and unsupervised learning. So when we say supervised learning, and we have some data from let's say animals, and where we know some of them are birds, some of them are not birds. So how we are going to train them, when we are not telling this explicitly to the program that there would be birds, there would be no birds, we just wanted to see how many different groups are there. So that's called unsupervised learning, that you see on the right hand side here. So now you see all of the birds are kind of picked as one group, that's being the clustering is working. And the kind of animals, the other animals, which will be reptiles or mammals, are picked into different groups. So this is called unsupervised learning, when the data is divided into certain groups without being shown group in the input sample. So let me rephrase it. So without providing program that there are these many groups, based upon the underlying patterns, how many group it is going to return. So that is unsupervised learning. In supervised learning, we force it to divide into certain groups. So let's say we say out of these many animals, these are duck, and these are the non-duck. So try to learn a pattern which can classify it, the ducks from the non-ducks. And whenever we have a new sample here, is going to put in one of these classes. So this is kind of difference between the supervised and unsupervised learning. And the reinforcement learning is kind of very advanced method in machine learning. Is here also we are not providing data labels, but what we are providing is an outcome at the endpoint. We are providing just the endpoint. The method needs to reach there. And for every kind of right step, it is being rewarded, and it's being kind of some score is given that kind of upgrade the model. And every time it is making wrong decision, it's being penalized so that we can lower the score. So ultimately, let's say we need to reach the score of 100. So every time it's making right steps, we say like, okay, add +1. And every time it's making some wrong steps, okay, penalize by 1 to -1. So to achieve it, the score of 100, every time it need to make correct steps, and so that it can jumps up from one correct step to the next and the next and the next. So ultimately it could reach the outcome or the endpoint. And this is one of the example, like realistic example of reinforcement learning. So when we are training dog for certain kind of activity, we are not giving any label to the dog, but what we are doing, let's say we wanted to throw some bone, and every time the dog picks up the bone and comes back to you, the dog is being rewarded. And in this case, you are observing the action of the dog for correct decisions, that, oh, the dog is picking the bone and bring back to me, or picking the ball, bring back to me. So it's being rewarded in that system. So the dog kind of learns. So every time I'm making this right decision, I'm getting some food or I'm getting some rewards. So this is a system of reinforcement learning. And this has been applied in many biological applications. In this case, this is kind of giving, when you wanted to reach there and you are making the right decisions, so there will be some score update according to your decision that you are making to reach your endpoint. And this is we already discussed. So now let's talk about the the method that we call as game changer. When a method will actually be a game changer, for that we need to have benchmarking. And what do we mean by benchmarking? Benchmarking is kind of applying all this method on a very independent data set. When we say independent, that means you know the outcome of that data, but this data has not been this data have not been seen by any of the method that you are testing. So in this case, the game changer method should qualify first that it's going to serve that purpose. Let's say you are interested in protein structure prediction. So first you will see all the qualifying methods, the methods which claim to be making protein structure prediction. Then you will pick all those methods, and then you will have a set of protein where you know the structure. So you know the structure now, but these structures are new, which were not available in literature before. So we assume none of these samples that are in independent data set are not seen by any of the methods for this purpose. So we're going to run all this method on this data set and see which methods is close enough to get the structure that we know. In this case, this is a benchmarking. You know the outcome. You know these are the methods you are going to test this data on, and then ultimately you see which method kind of gives you the closest result from the data that you already had. And this is what exactly been done in protein structure methods. And when we say game changer methods, so these are a few game changer methods that we are going to discuss one by one. The first example is from protein structure prediction. The next is from epitope prediction. The third is from identifying cancer subtypes. And the fourth is from data mining. And the fifth is from drug design. So I have only picked these 5 different methods based upon just my experience or my knowledge. So there are way more game changer methods, but we cannot cover all of them for the sake of time and for the sake of my limited expertise. So let's go one by one to the protein structure prediction. Going forward, for the first example for protein structure prediction, so AlphaFold is declared winner by catch competition. So let's talk about what catch competition is. The catch competition is kind of competition that's being started in 1994, I guess. And every year they have new structure submitted in the PDB, but it is not seen by all the methods. So they take out those structure and see there are many different methods which claim to predict protein structure. Which method is going to be the closest, give the closest score to the proteins that we already have in our data set. So and the AlphaFold, which is designed by DeepMind company owned by Google. So they say like proteins are the linear stretch of amino acid when they have different structure, like this is the secondary structure, and then this secondary structure fold into the seeds, this called tertiary structure, and ultimately they make some loops and turns to create their final structure. So this is the way that this works. Starting from the sequence, ultimately we need to achieve a structure that is similar to the protein that we already know. And when they have this a set of protein, solved by experimental method, and then they test all the different computational or AI method on those set of selected proteins. So, and then they ultimately rank those all those methods, and the results are published in this paper here. So they show the out of many different methods, they are not only showing 15 method, I guess here. So AlphaFold stands out to be the winner because it had the least RMSD, like root mean square deviation. So it had the least deviation of structure that they originally have. And if you can see the differences between the predicted and the experimental structure here, the experimental are in green and the predicted one are in blue. So they completely overlap each other, that you don't see any difference. That's why the RMSD, the root mean square deviation, was very minimum for AlphaFold. And these are only 3 different examples. Now you see these are overlaying the experimental method well enough, and this structure were very diverse. Now you see this structure is having only beta sheet, but this structure is way more complex, also has alpha helix and other terms here. So comparing all different structure, AlphaFold was declared as winner in 2020. 2020. And this is why we are calling as a game changer method, because this has a kind of one step closer to solve this problem of protein structure, which we are kind of struggling for so long to achieve this kind of accuracy. And the next kind of application that I have picked here is from epitope predictions. So before we talk about the methods, let's talk about what is an epitope. So epitope is a molecular, the smallest entity that is recognized by immune system. So our immune system recognizes certain stuff so that we can prepare our immune system to fight infections. And these epitopes are mostly kind of peptides coming out of proteins, and these are identified by experimental methods. There are many different assays to identify these epitopes, and all these epitopes are then kind of curated and stored in some databases. And one such example is IDB. So IDB has all these epitopes, and they are constantly updating this data. So every time there is new epitope, they are going to add it into their database, and this database is freely accessible. And even the epitope prediction, there are many different kind of arms, and the one of the arm is MHC class 1 binders. When we say like B cell epitope and the T cell epitope, in the T cell epitope, it could be further divided into MHC class 1 binder or MHC class 2 binder, or CD4 epitope or CD8 epitope for that region. So B cell epitope are very kind of simple. They kind of being recognized from the surface. So there is no intermediate partner between the antigen and the cell recognizing them, like the B cell. But in case of T cell, they are intermediate by MHC molecules, and this MHC molecule whom they are going to bind plays a critical role here. And this critical role is determined by the binding of those stretches or those peptides with the MHC allele is really important. And let's take an example how this process takes place. So whenever we have infection, let's say we have viral infection, the viruses are trying to integrate their DNA or RNA into the host, and then they ultimately secrete their viral proteins. This viral protein then chopped down into different fragments, and ultimately they create some peptides. This peptide then interacts with MHC molecule or specific MHC alleles and presented on the surface so that they can be recognized by specific T cells. So this is a processing pathway for MHC class 1. So if you look in the details of it, every step is very much in detail, and it could have several factors to determine whether we are going to have this epitope at this level, we are going to have epitope at this level, we are going to have epitope at this level. But this interaction seems to be the most significant one, which ultimately decide the fate of a peptide. So from this kind of pathway, there are many different tools developed, and I have picked one of the tool here for MHC class 1 binding, which is also hosted by IDB on this section here. And in IDB, as I said, they also have a database that is updated every week. So based upon on the new entries in the database, the weekly new entry in the database, they can run all the algorithm which claim to predict MHC class 1 peptides. So these are the different kind of server that they have implemented on, and these are the different runtime that they have run on this week, they have run on this week, they have run on this week, and then they score ultimately which method is giving most of our epitopes. Now you see one of the method by Denmark Technical University, NetMSC Pen, is consistently giving the best results, and that's why it has to be claimed to be a game changer. And the accuracy in terms of area under curve is one of the metrics to determine the accuracy, um, and the reliability of the method. The reliability of the method for MSc class 1 is super high. If I talk in the numbers, it is around 0.95 or something, or even above that value, and one could be the maximum. So it's very close to making very reliable prediction. So that's I have picked as the second method I'm calling as a game changer. The third method that I'm picking here is from identifying cancer subtypes. So when we say cancer as a disease, we are kind of summarizing so many different diseases into one. So cancer is kind of an umbrella term that covers so many different types of disease. And to understand it in the details, we try to divide it, whether it's a lung cancer, whether it's kind of a liver cancer or a pancreatic cancer or a breast cancer. We are trying to divide it based upon the tissues. So let's pick one of the tissue like brain cancers. Even when we talk about the brain cancer is not just one disease. It has many different kind of types of disease. They have a different survival outcome. They have different biomarker. They have different patterns associated with them. That's why, and they also have the different cell type that are being kind of affected by the disease. So let's say if the cancer is affecting one cell type, we are going to name it according to that cell type, and it's depending upon the cell type it's being used. So it can be either divided based upon the histology, the type of cell that being affected, or based upon the molecular patterns. So this is the underlying pattern, the molecular pattern. These are the mutations. These are the copy number changes. So there are many different molecular patterns that differentiate one disease from the another. So when we try to say for the brain tumor, so these are the different brain tumor types. So it could happen in the hemisphere, then we call it high grade glioma, ependymoma, or ATRT. It could happen into the pineal gland. Then there will be different types of cancer in the pineal gland. It could impact on the cerebellum, then it will be like a low grade or medullars, or again ATRTs. It could affect the like this part of the brain, like pons or spinal cord. Then again it will be high grade or ependymoma, or this could impact only the midline, and it could again be divided further into low grade, high grade, or choroid plexus tumors. So based upon what region they are impacting is one way to kind of classify them. The another way to classify them is based upon their molecular patterns, and this is what has been done by this group in Germany, DKFZ, and they this is the paper here. So they have taken all the different subtypes of brain tumors. These are the different brain tumor subtypes, and based upon their molecular pattern, which is determined by methylation array. So methylation array is a technique so give kind of a score for every methylated site in the genome. So there could be like 100,000 of methylation sites in the genome. So it's trying to cover most of them, and based upon the score that we are getting for methylation site, it try to find out whether we are close enough to classify a brain tumor in that is different from the another brain tumor. So if you can see the colors here, so there are many different types of brain tumors which were actually being kind of accepted as different brain tumor entities in by WHO. So this is kind of giving a summary of all the different brain tumor subtypes, and it has been very well accepted by World Health Organization, and so that we can treat them separately, we can kind of diagnose them separately. And this method that was published in 2018 is based on a random forest model from a reference data set, and ultimately they have selected this probe from this methylation array, and then they train their model, and ultimately after certain calibration, they could achieve a very decent performance. Now you can see these brain tumor types are actually kind of classifying in very closely related tumors. Let's say ATRT seems to be one tumor at certain point, but it says like even within the ATRT, which is one brain tumor subtype, there could be many different molecular patterns. Like it could be MYC, or it could be Sonic or it could be tyrosinase related. And these seems to be very much different from each other. And now you see this could classify even at the integrity of that level. And that's why I'm picking this another method as a game changer, because just based upon the methylation pattern, you can kind of classify what kind of brain tumor and a individual is having, and what level of treatment option we would be having for that tumor. And this is again based upon the supervised learning. The next thing I have picked here is based upon the natural language processing or text mining. Here, one of the paper that was published by this colleagues from Google. So they have developed a method called BERT, which is a bidirectional transformation language understanding. And to simplify it, it can understand the language, and it could kind of give the bidirectional response. So whatever you are asking, it can return certain response to that. So taking this kind of information, how we can apply into the biology and how we can apply that into our medical science. So this has been done by this method BioBERT. So they have taken all the paper from PubMed and PMC, and it is like billions of words here in those databases. So PubMed or this PMC PubMed Center resource are the center repository of the resources where you can see all the literature, all the publications. And then they also use this BERT, which is this method. So when they combine them together and they train all these keywords, and with the implementation from weight they have taken from the BERT method, they ultimately trying to understand and fine tune it to answer certain questions. So then they, to add it to the more context, they have also included some entities from disease databases, some relationship, and they start asking questions. So then they ask question, the adult renal failure causes. So then they have given options. So these are the options. What are the causes of renal failure? So it will give you an answer. So let me simplify it. Based upon all the PubMed records, a artificial method could answer your question. So while we are reading certain paper, we are reading it to get certain answer. Like we are trying to understand which protocol is going to be the best for my experiment. So how we are doing it? We are reading certain papers, and we see this method is going to be very suitable. So in future, you can do it just by supplying all those paper into a software, and then ask your question. So instead of you reading every paper one by one, this machine learning method BioBERT can read your paper and can answer your question. So in this case, they have asked variant of gene contribute to disease susceptibility, yes or no. So they have selected whether this variant is going to contribute to the disease susceptibility or not. So it could answer perfectly those details of questions. And let me rephrase this everything. This is one of the game changer method that I have picked and based upon the supervised learning. So but is added on a layer from the PubMed papers, and based on those paper, now you can specifically ask question, and the answer would be based upon all the paper that you have fed into this. And you can also customize this kind of text mining tool in near future, where you will supply the papers of your interest and then ask question that you are interested to know from those papers. So ultimately it's going to simplify your reading and giving you precise answer. The next method I'm picking here as a game changer is from drug design. This is based upon reinforcement learning. And in this is kind of a seminal paper where they're trying to uh, design inhibitor for a protein just based upon certain set of molecules. They said, we have this target, which molecule is going to be the best inhibitor in this molecule, in this chemical space. So they have picked many different databases, and from those databases they try to find out, we have a target which is a DDR1 kinase. For this DDR1 kinase, which molecule is the best inhibitor? So they have provided millions of molecule to train this, automatically train this model. And ultimately after 21 days, they come up with a model that is called generic tensorial reinforcement learning. That means this is a reinforcement learning method. They have not providing, these are the inhibitor, these are the non-inhibitor, this is the IC50 value, this is not, they're not providing anything. They are just providing the set of chemicals, and then ask computer which chemical is the best inhibitor for this molecule. And after this training they completed, it took 21 days to give you four hits. And when they tested these 4 hits, they were active biochemically, and out of them they also validated 2 of them in cell-based array. So out of 4, 2 were working in the cell-based array, and 1 was also tested in the mice, and that was working very well. So in a total stretch of around 1.5 months, they can design a molecule against a well-known target for fibrosis and many other diseases. So they have taken one target, and from the set of molecule, just based upon the reinforcement learning, they could design a molecule. And this process, when we do in practice, it was taking years and years, and they could stretch it to just 46 days and also have a very active molecule at the end. And this is another game changer method that I'm picking. All right, so now we talked about many methods and how these are the game changers. So why do some methods stands out? Why these methods are working and others might not be that good enough? And how we can also have our own method that could be a game changer. So all the game changer method have something in common, and I'm summarizing that commonality here. The commonality is most of them are based on the largest data set. So when you have large data set to train certain model, you are more reliably learning the patterns that out of that input feature. And like this NetMSC Pen, NetMSC Pen is performing very good because they try to kind of improve this method over and over again, almost in every years or in 2 years. So kind of like constantly updating. And the third thing that most of these methods are doing is feeding the model with the right feature set. Let's say for this brain tumor classification, they are picking the right feature set that is coming out of the whole genome instead of focusing on a small stretch. So when they have the whole genome and then they have selected, these are the probes that are contributing to this pattern. So picking the right feature is another kind of important thing that they are considering to stand out. And the third thing that they are considering is adopting a very different strategy. So in this case, the reinforcement learning for the drug design is a very different strategy. So they are also based upon reinforcement learning here, and that turned out to be to the new strategy to apply to this space. And the another kind of the key term that they are having is learn and relearn. So they constantly kind of updating, they constantly kind of learning this pattern and see these are the things that we are missing out and how we can incorporate these missing out samples in our future update. So those kind of learning and relearning is really important for a method to stand out. So let me summarize. We talked about these 5 different methods. The first one was coming from protein structure prediction, that is AlphaFold. The next one is from epitope prediction. So in epitope prediction is a big space. The one space here is MHC class 1 binder, and the net MHC pen method by Denmark Technical University really stands out in many many benchmarking results. Identifying cancer subtypes. So it could be done for many different cancer types. Like people are doing it for breast cancer, people are doing it for leukemia, people are doing it for liver cancer or pancreatic cancer. But I'm picking an example from brain cancer, and the model that was kind of really standing out is from the DKFC, the random forest model. And the fourth one I'm picking here is from the data mining. The data mining is the BERT method given developed by Google. And on the top of that layer, learning this example from PubMed is BioBERT, where you can supply your set of papers and ultimately you can get your question answered. And the fifth one we have picked is from drug design. This is a method based on reinforcement learning and trying to find out inhibitor against a DDR1 kinase protein. So these are the 5 methods that I'm calling as a game changer. There could be way more than that, but I could only summarize 5 of them. And so these are the common steps that we follow when we talk about machine learning method. The first is our input data. What should be our input data? So we cannot directly pass our kind of examples as such. When we have like inhibitor, we cannot supply inhibitor as such. We need to retrieve, or we need to convert those molecules into numbers so that we can train our model. And there are many different ways to convert your data into numbers so that we can feed it into this machine learning models. And then we need, the next step here is to split data. So if you are training upon all the data, how we are going to make sure our model is reliable? So to make it more reliable, we need to split data and train on a small data set and then test on the another small data set. So it's kind of a strategy to make you more confident on your data split and on your model. The third thing is model building. We have many choices when we are talking about model building. We can choose one algorithm over the other, and every time it's better to optimize or kind of go through many different iteration to picking the parameter, to fine tune the parameter, or to picking the actual algorithm that works very according to your data set. The next step is model evaluation. How we confidently are we going to say our model is working? Because we need to test and many different ways. We need to test based upon the internal cross validation. We need to test upon the external cross validation. And there are certain matrices which can help us to give a number that our model is reliable at this level, our model is reliable at this level. And the next is reusable models. So how we are going to, once we have done all this exercise, how we are going to save this model so that we can use it in our future projects instead of learning and relearning every time. So those are reusable model. So finally we can conclude our model with saving them, and so that we can apply when we see new examples are coming in. So that's all from my side. With that, I'm thank you for this and Dollar Education to giving this opportunity to present this understanding of machine learning to your audience. And feel free to ask a question or connect me over these sites. Thank you.
Thank you so much, sir, for this wonderful session. I hope the students have received much knowledge about the applications of machine learning and the steps needs to be followed. So we have some questions in the chat box. I'll read for you. The first question is from Santosh Basnet. He's asking, what is the difference between AlphaFold and homology modeling? Can we take PDB from AlphaFold 2 to design drug as like from RCSB bank?
Can we take PDB from AlphaFold to design? Yes, I think as shown in many of these benchmarking results, that AlphaFold is really standing out and is making predictions that are very reliable. So you can use those coordinates to design inhibitors. And this is only best when if there is a structure already available in the database like PDB, then it's better to go for the PDB structure instead of going for AlphaFold structure.
So we have one more question from Arpita Priyadarshini. She's asking if we are using IEDB and the epitopes are predicted, and by taking this its advantage, we can design vaccines also, which will be peptide based.
Yes, Arpita, that's a good question, and I think IDB is really contributing very well into this space. They have provided all this method free of cost for academic users, and you can use all this prediction. And they also have a database. From the database, you can go through all the known literature, and accordingly, you can decide whether you want to predict or it's already been shown in the literature. And then yes, this tool could be used to design vaccines.
So the next question is BioBot freely available, like it's a open source?
I think BERT is for open source, but I have not seen BioBot is free or open source. That's a good question. I need to check on that. So it's been published in BioNTech, so I'm assuming it is going to be open source.
Okay, participants, if you have any more questions, you can write in the chat box, or you can also switch on your mic and directly ask sir about queries.
Yeah, I think participants are kind of okay. They are not having any more questions. So you can go on with your stuff.
Okay, sir.
Yeah.
So the next question Monica is asking, can you please suggest some book for beginners in machine learning?
I'm not sure about just one book, but we can like having a workshop here coming in that I think Divya is going to talk about. So for the full core that Fernando is asking or the Monica is asking, we are going to discuss all this detail or all these methods, how you can build your own in our upcoming workshop.
So everyone, we are going to have a data science and machine learning in biology workshop from February 14 to 24. And the panel members for our upcoming workshop are Dr. Sandeep Dhanda, who is a bioinformatics research scientist at St. Jude Children's Research Hospital, Memphis, USA. Second, we have Ms. Dilraj Kaur, who is a research fellow in Indraprasth Institute of Information Technology, Delhi. And then we have Dr. Sherry Bhala, who is in consultant at Mount Sinai Health System, New York, USA. This is a workshop schedule. We have 3 sessions per week and 2 sessions in our last week. In the first week, we are going to discuss about the Python programming, handling data with Python, data clustering. Then we're going to discuss about in the second week, the dimensionality reduction, feature selection and data split, and building the machine models. And in last week, we are going to have model evaluations, and we'll be discussing about the case studies. And these are some feedbacks from our last workshop that we have organized last month. So we have received good feedback responses from our participants. And for this, you can like register yourself with on our website, that is www.dollareducation.org, and it's just a 2-step procedure. You just have to fill your basic information like your affiliation, your phone number, email ID, name, and then it will direct you to the payment gateway, and then after the payment you can directly register yourself.
Yeah, I think Monica is having question about, do we need Python basic knowledge? I think we are going to cover and start from the Python knowledge, how we are going to build those machine learning models. So we will start with the Python basics in that workshop.
If anyone is having any other question, you can ask directly.
So I'm not sure how we are going to take questions from the YouTube, or is there any question on there? I'm not sure about it.
Sir, I was checking on YouTube. I guess there are no questions till now.
All right. All right. So if there are no more questions, so we can end this session. And thank you all the participants, and hope to see you in this workshop. Thank you all.